Inside every living thing, there is a hidden set of instructions, a microscopic language dictating how a cell grows, divides, and functions. For thousands of years, early humans manipulated this system without sight. We altered the natural world through pure trial and error, never seeing the code we changed. Early farmers practiced selective breeding, choosing the strongest plants, and pairing the healthiest livestock to improve the herd. They recognized that favorable traits passed from parent to offspring, but the exact biological mechanics driving those changes were unknown. Early civilization relied on trial and error to shape the world. While we could see the results of breeding, the internal mechanics of life remained inaccessible, a system we could observe but never directly influence. By the 19th and early 20th centuries, scientists began hunting for the physical location of those instructions. One of the most significant breakthroughs arrived in 1953, led by scientist James Watson, working alongside his dedicated research partner, the scientist Francis Crick. This physical model represents what they found, the double helix structure of DNA. This twisted ladder shape is the exact physical mechanism that stores genetic information. Locating this structure took genetics out of the realm of abstract theory. Humanity finally had a tangible information system we could map, study, and eventually interact with. By the 1970s, researchers realized that if DNA operates as a code, it can be edited. At the time, treating diabetes required harvesting insulin directly from the pancreases of pigs and cows, a slow and resource-heavy process. Scientists devised a mechanical solution. They isolated the human gene responsible for producing insulin and physically inserted it into the DNA of bacteria. Because bacteria multiply rapidly, these altered microscopic organisms acted as living factories, churning out exact copies of the human hormone. This process generated life-saving medicine at a massive industrial scale, securing a reliable supply for millions of patients. The Insulin Revolution proved we crossed a critical threshold. By successfully reprogramming bacteria to produce medicine, we transitioned into the role of active biological editors. These microscopic organisms are just one example of how modern biotechnology operates. Today, we harness similar systems to power vaccines, engineer hardier crops, and develop targeted cancer treatments. Now, the defining tool of this era is CRISPR. It acts as a set of molecular scissors that cleanly and precisely cut specific sequences of DNA, allowing scientists to edit genes directly. The immediate medical application is clear. Inherited genetic diseases, conditions we have tracked for centuries without a cure, can now potentially be edited out of a patient's DNA. Researchers are also developing extreme long-term capabilities, from growing functional artificial organs in a lab to treatments customized to your exact genetic profile. Our technological capability has outpaced the slow crawl of natural evolution. We have acquired the literal tools to engineer artificial life from the ground up. But possessing the tools is entirely different from knowing how to use them responsibly. We must now define the boundary between eliminating a genetic disease and assuming total control over the biological inheritance of our species. The conversation gets more complicated when we move past disease prevention and into the territory of designing specific physical or cognitive traits for future offspring. The subjects of this vast biological experiment are human beings. The genetic decisions made today will cascade through the diverse populations of our future. Our ability to read and rewrite DNA has already secured our survival in ways early humans could never have imagined. But the moral reckoning over how we use that